Gene regulation is a process in which a cell increases or decreases the production of a specific gene product, such as protein or RNA. The main goal of gene regulation therapy is to introduce therapeutic genes into target cells. This leads to the stable expression of the therapeutic molecules and minimizes any inflammatory or toxic side effects. All domains of life use positive regulation, negative regulation, and co-regulation. Positive regulation turns on gene expression.
Clap on.
Negative regulation turns off gene expression.
Clap off.
Co-regulation turns multiple genes on and off to control gene expression. Gene regulation therapy occurs by inserting vectors, which are genetically engineered retroviruses, into cells. This vector overrides the faulty mutation, which leads to the reproduction of treated cells. Three main gene-based therapeutic approaches are being utilized as therapy today. Gene transfer gene-based therapy approaches and delivers mutated genes. Diseases caused by a lack of recessive functions in a single cell undergo gene transfer. Adding back wild-type genes back into the cells restores normal functions and alleviates the phenotype of the disease. Ex vivo and in vivo are both forms of gene transfer therapy. Ex vivo therapy approaches can be removed from a patient, be genetically altered, and then delivered back into the patient. In vivo therapy approaches deliver wild-type genes directly into a patient's affected cells, tissues, or organs. RNA modification gene therapy targets mRNA-encoded mutant genes. Dominant mutations in genes encode hyperactive proteins. Researchers prefer to inhibit mutant proteins with RNA approaches. RNA is targeted from the dominant negative gene and is then transcribed. This process inhibits the expression of the mutant proteins. Stem cell therapy uses human stem cells to repair disease-damaged tissue. Stem cells are immature cells that have not yet specialized and have the capacity to mature into various tissue types. Embryonic stem cells are derived from an embryo and are capable of unrestricted cell divisions. Ethical and technical issues have restrained the clinical use of embryonic stem cells. Somatic stem cells divide based upon the tissue type they are derived from. Although gene regulation can be controversial, it is important to realize that doctors were already treating hereditary forms of human diseases and metabolic disorders. CRISPR, first existing in 1987, has been a revolutionary tool in the field of the human genome. CRISPR is a technological system that is able to track down and edit genes by altering the genetic sequences of living organisms. With CRISPR, researchers can identify genes that cause diseases. In order to change genes, researchers have to track down the genes within a genome. The CRISPR system is able to track down and edit genes in plants, bacteria, yeast, and all organisms that possess DNA. In comparison to CRISPR, bacteria has a way to track down genes because they have been battling viruses for billions of years. Some bacteria have developed immune systems that record segments of viral DNA within their own genome. Enzymes in bacteria are able to recognize and cut viruses. This process prevents viruses from replicating. CRISPR makes altering the genes of plants and animals easier as time and scientific discoveries progress. With the help of CRISPR, scientists have discovered ways that piglets can provide livers, hearts, and other organs for humans. Researchers have also been able to make mosquitoes become resistant to malaria with CRISPR. People are actually able to purchase do-it-yourself CRISPR kits online. Therapies versus treatments have become a controversial scientific topic, for therapy treats diseases while treatments give advantages to individuals who are already healthy. Where do we as humanity draw the line? Are editing diseases out of future generations ethical? Diseases such as sickle cell, HIV, and certain cancers are a few diseases that can be cured by gene regulation therapy. Mutated cells can be removed from the body, edited outside of the patient's body, and then returned. Doctors were recently able to extract bone marrow from children who were diagnosed with a defective immune system and brain disorder. The mutated genes found in the bone marrow stem cells were injected with a genetically modified repaired gene, and repaired cells started to replicate. Gene therapy has also been used to cure type 1 diabetes, a condition in which there is little to no insulin production in the pancreas in dogs. Glucose and an enzyme were injected into the bones of dogs as a form of gene therapy. The distinguishing line between therapy and enhancement has become more questionable over the years because not everyone agrees on which genetic conditions need fixing. Although gene regulation can sometimes be considered controversial, various medical enhancements and discoveries have and will progress over the years.